Okay. Very good morning, my dear friends. This is Dr. Padala Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal. Today's our eminent resource person, Dr. Rani K. Madam Garu, Assistant Professor, Department of Botany, GDC, SIDDIPET Autonomous. Madam is going to explain an embryology area, plant embryology. So embryo sac, one of the important topic in our CPGET. So my dear friends, during this class, if you have any doubt, please ask our respected Rani Madam Garu. Madam will clarify your doubts and listen carefully, my dear friends. Okay, Madam, please start your session.
Students, today we are going to discuss about the megasporangium. So introduction, almost last class we'll discuss the introduction part of the female gametophyte. Today just the structure of ovule. I'm starting with the female gametophyte or the structure of the ovule. So almost an ovule or megasporangium, it develops from the base or the inner surface of the ovary. So this is the diagram, Nana. Almost the ovule or the megasporangium or female gametophyte. Anyone? So it develops from the base of the inner surface of the ovary. It is a small, generally oval structure, and it consists chiefly of a central body of tissue. So that is the nucellus. This is the nucellus. So, and one or two integuments also there. So each ovule that is attached in the placenta by a small stalk that is called funiculus. So this is the funiculus, stalk-like structure. That is the funiculus. The place of the attachment of the stalk with the main body of the ovule, that is called hilum. So almost this is the hilum part. Okay. So in an inverted ovule, the funiculus, it fuses with the main body of the ovule, it forming a sort of ridge. So that is known as a raphe. Almost this is the raphe. And the upper end of the raphe, it is the which is the junction of the integuments and the nucellus, that is called collagen. So this part is the collagen. Okay. So the nucellus, it makes the main body of the ovule, so which is made up of parenchyma tissue. And the nucellus is the megasporangium proper. So the nucellus, this is the megasporangium of the proper, and it is surrounded by two coats, that is the integuments. So this almost 1 and 2 parts, that is the integuments. Okay. Nucellus, it is made up of a parenchymatous tissue, and it is surrounded by two coats. So that is the integuments. In some Compositae and a few other families of the Gamopetalae, so there is only one integument, Nana, only Compositae family and some other families. So it's as per Bentham Hooker classification, Gamopetalae families. So there is only have one integuments. And in other plants like Santalum, Loranthaceae, so they are the parasitic plants, there is no integuments. And a small opening is left at the apex part of the integuments. So that is called micropyle. This is the almost opening place of the place, that is the micropyle. Okay. Small opening, it is left at the apex part of the integuments. So that is called micropyle. Where there are two integuments, then the interintegument is formed first and followed by the formation of the outer integument. Okay. So a large oval cell, it is lying embedded in the nucellus towards the micropylar end, that is the complete embryo sac. So this is the next topic is the embryo sac development, the round oval shape of the structure, that is the embryo sac. Okay. It's look like a large oval cell, it lying embedded in the nucellus and towards the micropylar end, that is called the embryo sac. Okay, so this makes the most important part of the mature ovule. Almost embryo sac is the most important part of the mature ovule. It is the embryo sac. It bears the embryo later on. Almost, so that is the female gametophyte. This, and next we'll coming to the structure of the ovule. That means the embryo sac. Okay, so just to you can observe this diagram only. The structure of the ovule. Integuments usually the ovule possess the either 1 or 2. That means unitegmic or bitegmic. Integuments also there. There is much variation among families as regard to the presence of the 2 integuments. Okay, so the number that is constant in most 2 families, and rarely unitegmic and bitegmic ovules, they will occur in same family also. Okay, so this is the complete the structure of the ovule. Almost the upper part is the antipodals, and 2 antipodals also there. So the female gametophyte and embryo sac, that is the 7-celled and 8 nucleate stage. 8 nucleate, the development of the embryo sac is of polygonum type. So this is completely called as polygonum type. And the megaspore mother cell, that means this is the megaspore mother cell with the megasporangium ovule. So it will undergo the 2 divisions to gives rise to a linear tetrad of the megaspores. So just to, this is this one, Nana, almost only just to will treat it as a double X 2n or 2n state of the female gametophyte. It will undergo the meiotic 2 meiotic divisions to gives rise to a linear tetrad of the megaspores. So normally one of the megaspore is out of this situated towards the collagel, the lower one, and remaining one is the going to the micropylar, towards the micropylar end. Okay, so just the functional megaspore, it represents the first cell of the female gametophyte, mostly collagel and just to, okay, this almost last class we will discuss some points. Mostly collagel and grows along the micropylar and collagel axis. So the nucleus undergo the 3 mitotic divisions and form the 8 nuclei. These nuclei are recognized 1 nucleus from each group at a pole. So it migrates to the center of the cell, that is called polar nuclei. So one will go the upper side and one will go the lower side. So the 3 nuclei left at the collagel end, and they are surrounded by the walls, and from the form the antipodals. So almost this 1 cell only, we will treat that the 2n state of the represent the first cell of the female gametophyte, Nana. It will undergo the 2 meiotic divisions. It gives rise the 4 cells. So almost in that one will functional and remaining. So that will undergo the 3 mitotic divisions. So in that 3, almost 1, 2, 4, 8 cells. In that 8, 3 will treated as antipodal cells, and remaining 3, so 2 are the synergids and 1 is the egg cell. Remaining 2 are the secondary cell of the nucleus. Okay, so this is also other diagram. So the structure of the typical ovule, that's look like anatrophous ovule. So it prior to the fertilization. Sac. Okay, the embryo sac in angiosperms is 8 nucleate and 7-celled. On the basis of the number of megaspore nuclei, so which take part in the embryo sac development, that recognized as 3 types. In that one is the monosporic, bisporic, and tetrasporic. Okay, so each type has more than 1 subtypes. Some of the common ones are the discussed continuously. So this is the types of female gametophytes: monosporic, bisporic, and tetrasporic. So monosporic, it divided into 2 subtypes, Nana. in that first one is the polygonum, so it will disappear. And second one is the enothera. In monosporic, two subtypes are there. So in that one is the polygonum and second one is the enothera. And remaining bisporic, so it is also two subtypes. In that one is Elium type and second one is the Endomion type. And next tetrasporic. So in the tetrasporic, almost in 7 subtypes are there. Peperomia type. So in that Peperomia, Pinia, Plumbago, Adaxa, Drusa, Fritillaria, and Plumbagella. So these 7 belongs to the tetrasporic female gametophyte or tetrasporic embryo sacs. Just we will discuss one by one. So this is complete overall development of the diagrammatic way. So in that monosporic, bisporic and tetrasporic. MMC ante generally megaspore mother cell. So undergo the meiotic division, meiosis 1 and second meiosis, and thus to ultimately the functional megaspore is 1 in the monosporic type and functional megaspore is 2 in the bisporic type, and functional megaspores 4 are involved in the development of the complete embryo. So that is the tetrasporic type. Okay, monosporic, bisporic and tetrasporic. So we will discuss one by one. In the monosporic, almost embryo sac is derived from a single haploid megaspore. So you know that almost this is the one cell. The microspore mother cell, this is common for all meiotic 1 and meiotic 2, but the arrangement is different. So in the monosporic, almost embryo sac is derived from a single haploid megaspore. So all the nuclei in the embryo sac are genetically identical. So as they develop from the same megaspore. There are 2 subtypes, polygonum and enothera. So this is the polygonum type of embryo sac. So just you can observe keenly, almost only the development part is different in polygonum and enothera. Just one is different, remaining all the pattern is same. So in this type, in polygonum type, almost the 8 nucleate megaspore type of the embryo sac. So this is first discovered by Strasburger Nana. Polygonum type embryo sac is discovered by first time Strasburger. In which plant? Polygonum divaricatum. So that is the polygonum type. 8 nucleate monosporic type of the embryo sac was first discovered by Strasburger in Polygonum divaricatum. So that's why it is referred to as a polygonum type. Okay. So it is common in angiosperms. Almost 90% of the angiospermic plants, it will show as this type only. So that's why it is the common in angiosperms. That's why. So it is normal type of the megaspore. So generally in this megaspore, the megaspore mother cell, only one is functional. So this is megaspore mother cell, Nana. The 4 megaspore formed from the megaspore mother cell. Only one is the functional, and that is participated in the development of the embryo sac. So this is the functional. Megaspore mother cell undergo the mitotic. So 1 will give 2, 2 to 4. In that 4, so upper 3 is the degenerated. Only lower 1, that is the functional megaspore. Okay. So this is the, it is located at the collagel place. So that's why the collagel megaspore is functional, and the remaining 3 towards the micropylar end, then 3 will degenerate. So the functional megaspore nucleus, it divide mitotically into 2 nuclei. So just we will take only this one, undergo the 3rd mitotic division. So it will undergo the 1 will 2 cells later. So as a result of 2 successive nuclear divisions, 4 nuclei are produced at each end. So division 4, division 5, 1 will give the 2, 2 to 4, 4 to 8. So in that 8, each, so after 4 nuclei are produced at each end. So in the 3 nuclei at the micropylar end, it form into egg apparatus. So this 4, this 4. In the 3 is treated as antipodal cells, and 1 will come to the center part. So below 3, so this is the almost egg apparatus, and in the egg apparatus, egg is located at the center, surrounded by the 2 synergids. So this synergid, this is the egg cell. Okay. So in the collagen end also, plasma membrane are formed. So it is surrounding 3 nuclei. They are known as a antipodals. These 3 antipodals, 2 synergids, 1 egg cell. So each one come for the center part. So 2 free polar nuclei, they from each end, they will fuse together in the central region to give rise to a secondary nucleus. Okay. So in this way, 7-celled embryo sac is formed. That is the polygonum type. So this is very common in angiosperms. It is normal type of the development of the embryo sac, Nana. So almost in the polygonum, which one is functional? Collagen megaspore only functional, remaining. So towards the micropylar 3, haploids are the degenerated. So the functional megaspore will undergo the other 3 mitotical divisions. So at the finally 8 nuclei are formed in the embryo. So in the 8 arrangement, 3 is the antipodal cells, 3 will come to the lower side. So in the 2 is treated as a synergids, 1 egg cell, and so each end almost the each in the collagen end as are the 2 prepolar nuclei from each end. So they will fuse together in the central region. It gives rise to a secondary nucleus. So in that way, 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleus, but in the cell formation 1, 2, 3, 4, 5, 6. So it will treated as a single cell. That is the 7-celled embryo sac is formed. So that is the polygonum type. So it is first discovered by Strasburger. In which plant? Polygonum divaricatum. Okay. And the second one is the Enothera type. So as almost 99% all the same except the functional megaspore. Okay. So in this almost the 4 nuclei to monosporic type of the embryo sac, it is first studied in Enothera lamarckiana. So which plant? Enothera lamarckiana by Geets. Almost so the polygonum is Strasburger and Enothera is the Geets. So hence it is known as a Enothera type. Enothera lamarckiana, and in this the micropylar megaspore that is functional and it take part in the development of the embryo sac. Almost manaki so in polygonum the collagen megaspore is functional, but in the Enothera type only which one is the functional, Nana? Micropylar megaspore. So 1 will come the 2, 2 to 4. In that 4, the lower 3, that means the collagen 3 is the degenerated, only the towards the micropylar megaspore. Haploid is the functional. So in that way, the micropylar megaspore is functional and it takes part in the development of the embryo sac. So the megaspore nucleus, it will undergo the only free nuclear divisions, division 3, division 4. It gives rise to a 4 nuclei which remain in the micropylar part. So in the polygonum almost other 4 is formed after 3 mitotic divisions, but in the Enothera after 2 mitotic divisions it gives rise the only 4-celled state of the embryo is formed. So in that with the almost undergo the only free nuclear, 2 free nuclear divisions to gives rise to the 4 nuclei which remaining in the micropylar part. So almost these 3 nuclei out of 4, 1, 2, 3, 4. So in that 4, 3 nuclei is formed as a egg apparatus, 2 synergids, 1 egg cell, and 1 nuclei is left as an organized polar nucleus. So it represents the uninucleate central nucleus. So the antipodal cells are absent in this type of the embryo sac. That is the very important in entrance point. Almost other only 2 mitotic divisions will take part. So one will formed the 1 to 2, 2 to 4. In that 4, 3 is formed as egg apparatus. One is the only uninucleate central nucleus is left in the center part. So in this, almost the antipodal cells are absent in the Enothera type. And next, the bisporic type. So in this type, the embryo sac consists of 8 nucleus, Nana. So as a result of 1 meiotic division in the megaspore mother cell, 2 cells are formed. So these are known as a dyads. So almost this one is the ileum. So in the plant bearing, bisporic in the first division is accomplished by wall formation, so that a dyad is formed. One of the dyad cell undergo the sec division, so whereas the other one is the degenerated. So both the megaspore, they will contribute in the formation of the mature bisporic egg cell. So each megaspore nucleus undergo the 2 mitotic division, it will form in the 8 nuclei. So the final organization of the embryo sac is similar to the polygonum type. Almost, so the bisporic embryo sac is classified into 2 types. In that, first is the ileum type and the second one is the endomeon type. So this is the ileum type of the bisporic type. So almost this is megaspore mother cell. Okay. So the ileum type also first described by Strasburger. Okay. Strasburger described the development of bisporic type of the embryo sac in ileum fistulosum. So why it is known as ileum means the which plant? The type of the plant is ileum fistulosum. So that's why it is referred as a ileum type. The megaspore mother cell. So this is the megaspore mother cell undergo the meiotic 1 division, 2 cellular form. So this is known as a dyad. 2 cellular, almost it is known as a dyad. So the collagen dyad cell become the functional. So this is the megaspore mother cell. It will undergo the first meiotic division, 2 cellular formed. So that is dyad state, the collagen dyad. So the lower one, collagen dyad cell become the functional and remaining micropylar dyad is degenerate. So it becomes functional and undergo the second meiotic division to gives rise to 2 haploid nucleus. So in this only the collagen cell after divisions, it gives rise to the 2 haploid nuclei. So both the megaspore nuclei take part in the embryo sac development. That's why that is known as a bisporic type. So in this almost one of the nucleus migrate to the micropylar. In this 2, one will each nucleus. So both the megaspore nuclei take part in the development of the embryo sac. One of the nucleus migrate to the micropylar end and other to the collagen end. So one micropylar and one collagen end. So in this 2 mitotic, after 2 mitotic divisions in each nucleus with the result of 4 nucleus. Okay, so this one. This is the almost other if you. So almost this one, Nana, one will to ఇక్కడ మన కలాజలే అంటే ఇక్కడ ఉండేటటువంటిది కలాజల్ హ్యాప్లాయిడ్ ఓన్లీ ఫంక్షనల్ రిమైనింగ్ 3 is the degenerated. So it will undergo the after 2 mitotic divisions 4 nuclei are formed at each end. So almost each end the egg apparatus consists of the 2 synergids and 1 egg cell towards the micropylar end and 3 antipodals to the collagen end. So the 2 free polar nuclei fuse together to gives rise to a secondary nucleus. So in this 4, almost 3 is antipodals, and 3 is 2 synergids, 1 egg cell, and remaining 2 free polar nuclei fused together to giving rise to a secondary nucleus. So almost this is the so this is the LEM type. Diagram, just this diagram is reverse, I think. This is the only collagel will functional and micropilar is the degenerated. So in this, it is known as a dyad. In that, one will go to the 4, 8 cells. In the 8, 3 is treated as antipodals and 2 is synergids, 1 egg cell. In the both sides, one will come to the center part, that is the secondary nucleus. So that is the LEM. And endomeon type. So as per same, in the endomeon, the megaspore mother cell undergo the meiotic 1 division. 2 cellular are formed. So the micropilar dyad cell becomes functional. Ikkada diagrams only the second step is reverse undi. So in this, almost the micropilar dyad cell becomes functional and undergo the second mitotic divisions to gives rise to the 2 haploids. So both the megaspore nuclei take part in the development of the embryo sac. One of the nucleus migrate to the micropilar end and other will the collagel end. So 2 mitotic divisions follow in each nuclei with the result of 4 nuclei are formed at the each end. So the egg apparatus consists of 2 synergids and 1 egg cell towards the micropilar end, 3 antipodals to the collagel end. So the 2 free polar nuclei fuse together to giving rise to a secondary nucleus. That is the endomeon. Almost to end. Next is the tetrasporic type, Nana. So in the tetrasporic type, the nucleus of the megaspore mother cell undergo the meiotic division, 4 haploid. So in this 4 haploid nuclei are the formed. All these 4 nuclei take part in the development of the embryo sac. So that's why this is referred or this is known as a tetrasporic type. The 4 nuclei get arranged in the just to the arrangement are differ from one to another. So in this, there are 7 types of the tetrasporic embryo sacs are there. So Adaxa type, Plumbago, Pinia, Peperomia, Drusa, and other Fritillaria, Plumbagella. So this is another way, Chrysanthemum type, need not to say here, almost to the 7 types. Okay, just we will discuss one by one. So first one is the Adaxa type. Adaxa type. Almost the nucleus of the megaspore mother cell undergo the meiotic division and 4 haploid nuclei are formed. Okay, so the 2 nuclei shift to the micropilar end and the other 2 to the collagel end. Okay, in that 4, 2 will 2 is the collagel end and 2 is the micropilar end. So these nuclei undergo free nuclear divisions and at each pole, so 4 nuclei are formed. So this one and this one. One nucleus from each pole shift to the center of the embryo sac and it become the cellular. So there's a diploid secondary nucleus is formed in the embryo sac. The 3 nuclei at the micropilar end. So it becomes the cellular and form the egg apparatus. In the egg apparatus, so 3 nuclei at the collagel, 2 is the synergids and 1 is the egg cell. 3 is the antipodal cells. So it's look like a common type in the tetrasporic embryo sac. Adaxa. So Adaxa means 8 nuclei which are formed by the mitotic division of the 4 haploid nuclei. So it's look like a cenomegaspore. So the arrangement of the 8 nuclei in the embryo sac is the same as in the polygonum type. So example Adaxa, Sambucus, Almus, Tulipa, and Erythronium. These all are the example of the Adaxa. And Plumbago type. So in the Plumbago type, so this type of the embryo sac is characterized by the absence of synergids and antipodals. So you can observe these diagrams. Almost in this type, the embryo sac development, the nucleus of megaspore mother cell undergo the meiotic division and 4 haploid nuclei are formed. So same, the 4 nuclei gets arranged to the 4 sides. So 1, 2, 2, 2, 2, 4, 4 will arranged at the 4 sides. 1 micropylar, 1 collagen, and 2 is the opposite sides of the embryo. Okay, so the 4 nuclei get arranged in the 4 sides of the embryo sac after the formation of the vacuole. Manaki is generally, so in the center part, one vacuole is formed. So in that one, the 4 will dispatch to the 4 sides of the embryo sac. So the 4 nuclei undergo the free nuclear divisions. Pre-nuclear divisions. So 1 and 2 nuclei are formed at each side. 1 nuclei from each side, it will get arranged at the center. Almost 1 will undergo the 1 mitotic zone, 2, 2, 2, 2 is formed. So 4 will arranged singly 4 sides, and each one comes the center part of the embryo. So the 4 nuclei. Okay. So almost 1 nuclei from each side, it will get arranged at the center. It becomes the cellular, and the tetraploid secondary nucleus is formed. So in the tetrasporic, 4 nuclei is fused together to the center part. Okay. So the nuclei at the micropylar end forms the egg, and the remaining 3 nuclei becomes the cellular, and it will form the parietal cells are degenerated. Almost this will undergo the 2 mitotic divisions, 3 antipodals is formed, and this will also undergo the mitotic division. Either egg apparatus is formed. So otherwise, these 2 are also mingled with this side. So 2 is treated as a synergids, and 1 is the egg cell. So that is the plumbago. And pinia type. So in the pinia type, it is found in the Euphorbiaceae and Malpighiaceae family. Almost the Piniaceae, Malpighiaceae, and Euphorbiaceae. Okay. So in this, the 4 haploid nuclei of the Cenomegaspore undergo the 2 successive mitotic divisions, it forming the 16 nuclei. So you can observe here megaspore mother cell, after 1 division 2, 2nd division 4, 3rd division 8, and 4th division 16. Okay, so each 4, 4, 4, 4, 2, 1 is micropylar side, and 1 is the collagen side, and 2 is the sides, opposite sides of the 4, 4 cells are arranged in the embryo cell. So this is the pinio. The megaspore mother cell undergo the meiotic division and form the 4 nuclei. So the 4 nuclei undergo the 2 mitotic divisions and form the 16 nuclei. So these are arranged in 4 plus 4 plus 4 plus 4 manner. Almost 4 plus 4 plus 4 plus 4 manner. Okay. So at the 4 ends of the development of the embryo cell. In this each, that means, sorry. In this, so 1 nucleus from each group of the 4 nuclei moves to the center, where all the 4 nuclei fuse to form the tetraploid secondary nucleus. So this one, okay. Each group, each 4, 4, 4 cells, 1 cell come to the center part. So the 4 nuclei fuse to form the tetraploid secondary nucleus. Okay. So the 3 nuclei situated at micropylar end, it organized into the 2 synergids and egg cell, and 3 nuclei at the collagen end become the cellular and form as a antipodal cells. Okay. So remaining these 3 place. So the remaining 2 groups of the 3 cells, it form the triad set to both the sides. So after some time, it's degenerated and it will mingle with the cellular endosperm cells. Okay, so that is the pinia. The best example of the pinia type, Piniaceae, Malpighiaceae, and Euphorbiaceae. Okay, and peperomia type. So next one is the peperomia type. So in this, the egg apparatus, this type of embryo sac, it is first discovered by Campbell and some Johnson. Okay, the peperomia type embryo sac was discovered by Campbell and Johnson in peperomia plant. Peperomia plant. So the megaspore mother cell, as a result of mitotic division, it forms the 4 nuclei, these 4 nuclei undergo the 2 mitotic divisions and forms the 16 nuclei. So megaspore mother cell after 2 divisions, 4 nuclei, and after 4 nuclei undergo the 2 mitotic divisions, form the 16 nuclei. So in this 16 nuclei of these 2 nuclei at the micropylar end, diagram is just disappear. So the 2 nuclei at the micropylar end, it become the cellular and form the egg apparatus. So it consists of 1 egg cell and 1 synergids. So it's the 6 nuclei at the collagen end, it becomes the cellular and form the 6 antipodal cells. So the remaining 8 nuclei fuse to form the secondary nucleus at the center of the embryo sac. So it forms as a octet condition. That means 8-end state. So 8 nuclei almost in the 16, 2 at the micropylar and 3. So each is 2 cells come to the center part. So the almost 8 nuclei fuse to form in a secondary nucleus at the center of the embryo sac. It forms as a 8-end condition or octet condition. And the next one is the Drusa type. So in the Drusa type, it is also known as 3 + 2 + 11. So other way, almost it is in 16 nucleate embryo sac, Nana. So this type of embryo sac is found in Drusa optocitifolia. So it's Drusa type simply. This nucleus of the megaspore mother cell undergo the meiotic division and 4 haploid nuclei are formed. So out of this, 1 nuclei shift to the micropylar end and other 3 to the collagen end. So almost the diagram different. The nuclei at the poles undergoes the free nuclear divisions twice and 4 nuclei are formed at the micropylar end. So this is the micropylar end and 12 nuclei at the collagen end. Almost this is the almost micropylar, just we will treat it as a reverse. This is the micropylar, this is the collagen end. Just to in this, out of this 1 nuclei shift to the micropylar end, this one, and other 3 is the collagen end. So the nuclei at the poles undergoes the free nuclear divisions twice and 4 nuclei are formed at the micropylar end and 12 nuclei at the collagen end. So this is in this 1 nuclei from each end shift to the center and a diploid secondary nucleus is formed. These 2. Okay. So the 3 nuclei at the micropylar end becomes the cellular and forms the egg apparatus. Remaining 11 nuclei, so it becomes the cellular and form the antipodals at the collagen end. So for example of the Drusa, Rubia, Chrysanthemum, Almus, these all are the examples.